Thanks, Michael. Mark, could you please put the timer on hold? I've got 10 minutes. I've got some really great stuff that I need to show. Yeah. So apologies if I missed anyone. I know there's a lot of stuff in this presentation, and not everyone is acknowledged, and not all of the participants in the projects are acknowledged. So I apologise for that. So just a quick recap. Program 4 really is where a lot of the technologies are trying to be applied at field scale and for grower group outputs. And we've got essentially three key components in our program. One is around the novel plant and system-based approach for improving soil. The second output is kind of the work that Hassan was mentioning before, and Amanda as well, around some of the novel physico-chemical type intervention strategies for developing high-performing soil. And then we have the soil improvement decision support tools. So just some brief highlights from Program 4. We're across all the projects in Program 4, we're engaged with 15 grower groups, 8 unis, a whole stack of research partners. We've finished 3 projects so far, and we've currently got 9 contracted projects. And importantly, we've got one that's contracted with GRDC. So it's a co-funded project with GRDC. Which Nigel will describe soon, and also one that's recently been co-funded through DPIRD, Western Australian Carbon Futures. I think David Minke is leading that project, so yeah, please track David down to find more about that project. We've got some really exciting PhD students, and I'm going to present some of their work here, but I know some of it will be presented tomorrow as well. And after my talk, we've got, as I mentioned, some much anticipated outputs from both Nigel and Gwen Greelett on the regenerative farming systems and the role of carbon in those systems. So I think there's a lot of anticipation there around those outcomes. And importantly, last time I counted, we've got 18 field sites collectively. So it's a lot of field sites all around the country, from the very top of North Queensland right through to the bottom corner of Western Australia. So we've got a lot of geographical coverage with our field sites, and I think there are 3 additional ones that are currently being set up or planned at the moment. Okay, Cook's tour of our projects, and again, I'm going to have to rush through this because we've got a lot of stuff to cover. So Terry Rose has been leading one of our larger projects around plant-based solutions to improve soil performance. And we're just going to focus on a couple of the key sites here. So the Riverine Plains field site, about 3 years of cover cropping. And distilling everything down, we can see the more complex information there, but we just distill it down. Is it works when you've got water, and it doesn't work when you don't have water. And where you can grow a reasonable cover crop, good things happen. And I think, Terry, hopefully that's the key message from some of this work. And some of the good things included increased glomalin or protein content of soil, which is related to soil structure, which then also related to improved saturated hydraulic conductivity. So some really good sort of physical outcomes from that cover cropping. Another component of this work, please track down Luca, who's a fairly recent new member of these projects, and Richard Bell around some of the work in the Western Australian wheat belt. So I think we're familiar with some of the issues on the sandy soils. There are large tracts of land that are not farmable, that are just still farmed, but the farmer basically doesn't get much return from them. And we're looking at some fairly unique farming systems, including the use of Lebbeckia as a cover crop in those systems to improve the soils there. But please track down Luca to get some more details on that. Okay, Cameron Copeland, one of our PhD students, he has got some really interesting work around, and it's a bit of a chicken and egg story. So what controls the soil biology? Is it the biology that's present, or is it the soil type? And essentially what Cameron has done is he's taken the biology, taken the people out of the room in the soil, and then changed it with the microbiome from different soil types. And some of the early findings really are showing that it seems to be the soil that's controlling the biology. So he's put different microbiomes back into sterilised soil, and it kind of always reverts back to the same type of metabolomic profile, same type of microbiology. So some really interesting outcomes there, and what Cameron's trying to do now is trying to understand the role of that microbiology in wheat yield. or that change in microbiology of the wheat yield. So yeah, Cameron's right over here, chat with him, he's got a really interesting story. Hanlu, I haven't caught up with Hanlu yet, but similar to some of Terry's work around cover cropping, it comes down to the water. And a lot of Hanlu's work has been done in much higher rainfall situations where cover crops, in particular the summer cover crops, are much more reliable. We're talking 1,000 mil rainfall type zones. And essentially they're showing that, her PhD is showing that the cover crops are increasing soil carbon, increasing microbial functions, including mycorrhizal fungi. So again, the cover crops are having a great outcome for the soil, but it's very water dependent. Okay, Navita is another PhD, another very exciting project looking at sandy soils, and in particular, having a look at the role of manipulating the soil microbiome to assist in mitigating some of the issues with the non-wetting soils. So yeah, track down Navita, who can give you a great description of her PhD project. Now, there's been lots of chatter on resilience and resilient soils. What does it mean? And when I say lots of chatter, you go online, you'll find hundreds of articles talking about soil resilience or resilient farming systems. There's no metrics on it. So, what do you actually measure for a resilient soil? So, Zhang Liu from Griffith University is a PhD student, but there's a broader project with Professor Chen and Mick Rose looking at actually trying to develop metrics and evidence for systems that become more resilient. So we've got some really cool mechanisms and lab-type tests for measuring what a resilient soil will be, in particular microbial functional resilience to stresses like drying, drought or compaction. So track down Zhang Yu, and I think actually Zhang Yu's giving a talk later on today, so yeah, hopefully he'll explain some of his work in more detail. Okay, we get now more onto the chemical constraints of soil or the physical constraints of soil. Jason Condon has led a project which has just wrapped up now, mainly a lab-based study, trying to understand different amelioration strategies and how they might impact these multiple soil constraints. And there's some great outcomes from that, and I think Jason's also got a video on the Soil CRC webpage. Please go to the Soil CRC webpage, track down that video. It's 2.5 minutes, I think, and it's a great read or listen or whatever you want to call it. Ehsan. So we all know Ehsan, we all love Ehsan, and he's got a great field site here at Lockhart. And there's also a nice 2.5 minute video that Catherine and her team put together on that field site. So please go to the CRC webpage, track down that video. There are other field sites within this, including in Victoria and Western Australia. And Rob Miller will also be setting up a site up in the Burdekin looking at sodic dispersive subsoils in the sugarcane system. And there's been some really interesting data coming out of these field sites. I was having a chat with Shane, Wayne and Dave, the technical team from Wagga, and they've got the two plants. The one on the right is from the control plot, and the one on the left is from some of the subsoil amelioration. There's clearly a major change in biomass, but what's also really interesting is a change in the root physiology. So the control soil, where the roots actually have got a subsoil limitation, growing that way. Where we fix up the subsoil, the roots are growing that way. So they're definitely penetrating deeper into the soil, having much better access to resources. And by that I mean water and nutrients that have moved through the soil profile. So it's all about resource use efficiency, and I think we're going to be assessing that in more detail, and I'm very excited to look at the changes in particular in water use efficiency and how this crop will finish. Another important component in this crop, in this particular field site, is having a look at the role of these amelioration options on nitrogen fixation and also carbon allocation. Can we increase that carbon use efficiency? I think Terry asked a question before, what is carbon use efficiency? Our photosynthate, can we get more of that photosynthate being stabilised in soil aggregates at depth? And that will be studied in this trial as well. Alice Melland has taken over from John Bennett on the project assessing soil moisture and water use efficiency across our farming systems, and there's quite a few opportunities here, I think, for Alice to work some of our grower group field sites. So yeah, this project is only really just starting to kick off now, and I'm hoping that we can report on some of this work in a couple of years' time. Another very exciting PhD project, Davin. Please track Davin down later on. So everyone knows about pH, the H pluses in your soil. We all know how to ameliorate H pluses in soil by adding lime. What very few of us actually investigate is the E minuses. So for every H plus, there's an E minus in soil, a redox. And there's some very new opportunities around manipulating soil redox conditions. That's basically the amount of electrons in soil and how these can impact both nutrient uptake and also nutrient availability. And Davin's got some great data using sort of electro cabinets and electro methodologies where we're pumping electrons into the system, and he's actually showing phosphorus release as that iron. So a lot of the phosphorus in soil is tied up with iron, where you can reduce the iron from iron 3 to iron 2. You get a concomitant release of phosphate into soil solution. There's some very exciting data coming out from Davin's work, but please track him down, or Andrew Rose, and they can explain the whole process in a lot more detail. I didn't hear that, Mark, thank you. I'm actually nearly done. So Keith and Chloe have done a stack of work. This project is finished up. The report's on the CRC webpage, and they've done a whole stack of work updating a few of our models. So we all know and have used AppSim, some, like myself, far less than others. But Keith is going to give a demonstration tomorrow of some of these new model updates at the Program 4 meeting, I think at 10:15. So please come along to that and have a look at how these models, in particular around phosphorus, nitrogen volatilisation and water repellent soils, how those updates have been put into AppSim. And another PhD student project, Suman. So Keith is very excited about this, and I'm actually also quite excited. It's a little bit about re-engineering or having another look at how AppSim works and reverse engineering AppSim to understand what the soil constraints are. So wouldn't it be nice to put your yield data into AppSim, and that can then reflect what constraints are present in your soil rather than having to go out and measure your constraints by primary methodology. And then from that, we're hoping that can then feed into our future decision support systems, which will give you then information around the best ways to address those constraints and improve yield. That's everything. Well, not everything. I did actually miss Mick's project. So Mick's project around soil herbicides has just wrapped up, and the reason I didn't show it here is because he missed the deadline to give it to me. So he sent it last night after the pub, and I'd already sent my presentation to Catherine, so apologies for that, Mick. But yeah, please track Mick down. Got some really new and innovative tools for assessing herbicide, in particular pre-emergent herbicide residues in soil, and predicting plant-back issues for that. And we're looking at some possible collaboration down the track with GRDC on this, and looking at some commercial opportunities. So rather than, and it's a huge issue across Australia, pre-emergent herbicides not breaking down when the soils are particularly dry. How do you actually know if those residual herbicides are going to impact your crop itself? So we're starting to get some pretty solid tools on that. So track Mick down, he'll give you more information. Thank you.